Thanking everyone for sticking around until the end. Hello everyone. I am Mohan from Martin Kirch's lab at Berlin Institute of Health at Charité, Germany, and I'll be presenting our research titled Accelerating High-Throughput Characterization of Regulatory Variance with Deep Learning. I want to start with this schematic from Gasparini et al., which shows an enhancer in action, thereby where you see that this enhancer is trying to regulate its target gene. FYI, enhancers play a crucial role in regulation of gene expression, which underpins human development and homeostasis. And this picture also shows all kinds of biochemical annotations that we tend to use for nominating enhancers and can also utilize to map enhancer to their target genes. Now, though depicted in this 3D proximity quite conveniently, the mechanistic significance of this enhancer-promoter interaction is still an area of active investigation. And it has been also established that more than 90% of disease-associated single nucleotide variants are also located outside of the coding genome. Now, given the search space, identifying which of these non-coding variants are functional and how they drive phenotypic changes remains a major challenge. While epigenetic marks and TF binding can suggest candidate variants, their broad genomic presence makes pinpointing causal variants really difficult. This is why we need characterization of regulatory variants as much as possible. And but lucky for you, we have technologies just to do that. For example, I bring you here something like massively parallel reporter assays, or MPRA. In a typical MPRA, you start by having a candidate cis-regulatory element, or CRE, which can either be a genomic region or can also be a completely synthetic element. And it can also have certain variants in them or not. And initially, we tag them with certain barcodes, which makes the downstream analysis easier. And then we insert this segment into a plasmid, which contains important components for MPRA. And then we transfect this plasmid library to our cell type of interest by using a lentiviral or other kind of construct. And upon a certain incubation time, the RNA and DNA content is then extracted, sequenced, and then analyzed to compute regulatory activity for our candidate CRE. And if a variant is involved, then we can also get something like variant effect by comparing this alt regulatory activity to the reference sequence. Now, given the challenge and this technology in mind, we sought to ask two important questions. Mainly, how do these single nucleotide variants alter the landscape of cis-regulatory elements? And can we leverage MPRA data with machine learning to quantitatively predict variant effects? Now, we started off by selecting regions and variants for our MPRA testing. Now, unlike most studies which tend to focus on a specific loci or phenotype, we used regions that are in cis direction for all the genes that are in human genome and went until 20,000 bases and also avoided certain core proximal elements. And in a chunk size of 300 bases, we took the central 100 bases and performed in silico mutagenesis by adding more stimulated variants. And in our final design, we also used an in-house built machine learning classifier, which was initially trained to predict open chromatin, to rank this candidate CREs with variants. And we selected certain cell type specific and agnostic, and also some global positive, neutral, and negative variants for our MPRA testing design. This also included some universal controls. These are regions that are open in all cell types or closed as DNase controls. And we also included certain positive and negative variant controls from previous MPRA studies. Next, we performed our MPRA in two human diverse cell lines, namely the liver cancer cells, or HG2, and also the human embryonic kidney cells like HEC290T. And the results looked quite promising with a replicate correlation among the three replicates were about 72% for HG2. And more importantly, we also saw a significant difference between our previously included MPRA controls and also our newly developed DNase controls. And this trend was also observed in case of HEC290T, thereby reinforcing the robustness of the approach. Now, to understand the landscape of creeks better, we tried to identify variants of significant effect. And for this, we performed a differential expression analysis by using an in-house developed tool called BCOM for allelic comparison. And this tool allowed barcode level input and can also perform outlier removal given as an option. And ultimately, we identified significant upregulating and downregulating variants for both cell types, which are about 14% in case of FG2 and 10% in HEC290T, among the number of variants that we actually test after our barcode filters, and all at the false discovery rate of 10%. And now we took this significant variance and then used something like FIMO, which comes from MemeSuite, alongside the latest Hokomoko version 12 database to find motifs that this significant variance fall within. And this honestly opened a floodgate of many kind of scenarios, which we don't have time to go through all of them, but I bring you the one that has the agreement with the majority of cases, where we saw that for certain motifs, for example, like kappa and USF2, where the presence of the variant ends up creating this motif, and we can clearly see that it has a significant high effect. And we also found certain examples, for example, HNF1 beta or KLF16, where the presence of variant destroys that motif, and we saw a reverse effect of being negative fold change than the reference. Now, this suggests that presence of a variant will have an inverse effect in this context, but there were definitely some exceptions, which also suggest that there are additional mechanisms at play. Now, I just want to request you to keep these names in mind as they're going to come up later. Next, with our MPRA data, we went back to our initial deep learning classifier and fine-tuned it or reiterated it to have a regression output, in this case as a multi-class regressor. And this is a very simple design where the input is the nucleotide sequence, and it has few arrays of convolutional and batch normalization layer, and ultimately two dense heads that are supposed to predict cell type specific regulatory activity. And we used only our reference sequence library and went with a classical train validation test approach. And our best model could get a test Pearson correlation of 65% in case of FG2 and 63% for HEC290T. I can also admit that while the model's performance is not perfect, it is actually approaching the level of agreement that we saw earlier in the experimental replicates, which were about 70%. And this is generally considered as a good indicator of reliability in this context. Next, we went with the most obvious step, which was about finding out which features were important for activity. And for this, we use a popular tool called TF-Motisco Lite and use the same motif database that we used earlier. And this, as expected, we were able to recapitulate all general transposition factor binding sites, which are KLF16, NFYC, USF2, and RF1. And we found these in both the cell types. But more interestingly, we also found some liver-specific TF binding sites in case of FG2, which were HNF4 alpha, kappa, and HNF1b. Now, this definitely brought in more confidence to our modeling approach. So we next performed, we used our model to predict variant effect. Now I show you a more of a summary slide where we can see that our trained MPR NN was able to get a Pearson correlation of 36% in case of FG2 and 28% in HEC290T. For comparison, we also utilized two large deep learning models, which are namely Informer and Psi from DeepSea, which do not directly predict variant effect, but they claim to approximate variant effect by using certain cell type specific features. but they were performing quite poorly, which were about, on average, 22% in case of HepG2 and 16 to 17% with HEC. Now, clearly, MPR-NN has a bigger hand here, as it is directly trained on the same task, and others attempt to approximate this. But the real focus here should be that because Informer and Psi are trained for broader genomic epigenetic features, and they may not always translate or warrant variant effect. And I'm also glad that we talked about what model to use in which scenario on day one. So I just want to show that the large DNN models like Informer are computationally intensive and can take significant runtime, whereas smaller models can actually do it much faster, but are also limited to the usage. You can only predict for the cell types they are trained on. And this trade-off between speed and usage are actually important to consider when selecting a model for, at least for this task, which is predicting variant effect. Now, in summary, we performed an extensive MPRA on 174,000 variants across 63,000 regions in 2 diverse human cell lines. We used this data to train our model MPR-NN, which was able to predict variant effect at a Pearson correlation of 36%. There's definitely room for more improvement. Now, the model is also fast in training and also offers easy interpretability, where we also saw that it was able to capture some cell type specific TF binding sites, at least in case of FG2, but mostly the general TFs in both cell types. And we also studied the impact of SNVs in enriched motifs and could observe a subtle inverse effect. In future, we are looking to understand the relationship between cell type specific gene expression to function of Cree activity, and also the variants that they have within these Crees. And we want to investigate the impact of variant in regions on gene expression by mapping this to the gene TSS. And we are also trying to give a fair comparison to these large DNN models by approaching something like a stack modeling approach, which was done also in the past. And in the end, I want to thank Professor Martin Kircher and Max Schubach from Kircher Lab for their constant guidance, and a huge shout out to Nadab Aitub's lab, where we performed these MPRA experiments, and all other contributors to this project. Thank you for listening.
We have time for one more question while we set up for the next speaker. I have a question actually. So have you thought of, I really like the way that you fine-tuned on the task. Have you thought of using the embedding from Informer instead of like the one-time encoding, like the sequences that you use to kind of train your model?
Right.
So I guess in the model, you basically one-time encode the genomic sequence.
Right.
Presumably, you can take that same sequence, run through Informer, and grab and embed it, and do that into your model, fine-tuning on your experimental.
Yes, that's definitely a
Any thoughts on that? Do you think that would work well, or?
With Informer, we're not sure about doing that. But first of all, within Informer, it asks for a really large context, which are about some 100,000. It's quite big and also computationally intensive. And we have another model that we could actually use this strategy as well, which is 10 to the psi. It's much smaller and faster, and can actually get better embeddings as they have more output features, which are about 21,000 epigenetic profiles compared to 5,000 in case of Informer.
But that's TBD.
Yes.
Okay. Thank you. Can I ask you? Well, sure. Yeah,
So, Informer is known for not being able to predict personal genome variation and impact on expression, but the Katie Pollard recently had a preprint that's called Performer, which uses Informer but fine-tunes it on or trains it on the personal like genotype and matched expression. And then have you tried to look at like where your MPRA-NN would stand with respect to?
Not yet, but
Okay.
we will also take that into account and have a comparison. But I think those methods, I mean, our methods are still not genome level applicable.
I see. Only on them.
They're only mainly trained with enhancer sequences, tend to have a different DNA
Sure.
properties compared to other classes of genome. So something to get into.
I think it's